Have you ever looked at a towering redwood tree, stretching hundreds of feet into the air, and wondered how it shares a biological connection with the invisible, microscopic bacteria living in your own gut? The sheer variety of shapes, sizes, and traits in nature can look like random chaos, but to build something this complex, you can't just guess where the pieces go. You need a precise, universal instruction manual. That manual is hidden inside almost every single cell of your body. It's called DNA, a microscopic vault containing the exact blueprint required to build every organism on Earth. Understanding how life actually works requires us to decode this manual across 4 specific scales: the molecular letters it's written in, the proteins it builds, how it gets passed through generations, and finally, the way it organizes the entire planet. DNA is written in a biological alphabet called nucleotides. The latter's rungs are built by chemical pairings. Adenine locks with thymine. Cytosine pairs with guanine. In RNA, we see a key swap. RNA is single-stranded, completely replacing thymine with uracil. Every human cell contains about 2 meters of this code. To fit that data into a microscopic nucleus, the cell spools the genetic thread around specialized proteins called histones, packing it into the dense, X-shaped chromosomes you see in textbooks. This compact storage system is an elegant way to protect data. But stored instructions are useless without a mechanism to read them and build a physical body. This creates a logistical problem. The original DNA is far too precious to leave the nucleus, but the ribosomes, the cellular factories that actually build the body, are located outside in the cytoplasm. The cell solves this through transcription. An enzyme unzips the DNA to create a temporary, single-stranded mRNA photocopy. This allows the master blueprint to remain safely locked in the vault. Before leaving the nucleus, the photocopy is edited. Spliceosomes snip out non-coding introns, gluing the essential exons together. This edited strand travels to the ribosome for translation, reading the code in 3-letter codons. tRNAs deliver amino acid blocks, snapping them into a long chain. This assembly line, from DNA to RNA to protein, is the central dogma of biology. It is the precise physical process that transforms invisible molecular code into the complex 3D proteins that build every trait in a living body. Those protein-building instructions are passed down through generations. By studying thousands of pea plants, Gregor Mendel discovered that traits are governed by distinct alleles inherited from each parent. Mendel proved that alleles separate during reproduction, and dominant ones can mask recessive ones. As this Punnett square fills in, you can see how a dominant tall trait results in a physical 3-to-1 ratio, even when the underlying genetic code is different. But biology often bends these rules. Incomplete dominance can blend traits together, like a red and white flower producing pink offspring, while epistasis allows one gene to act as a mute button that blocks another. The interaction of these dominant, recessive, and masking rules is what generates the vast, observable physical differences between every individual on Earth. Accumulating inherited variations over millions of years results in the millions of distinct species today. To organize that planetary complexity, biologists use a universal filing system called taxonomy. This system starts at the highest level by splitting all life into 3 broad domains: bacteria, archaea, and eukarya. While bacteria and archaea are single-celled, they are genetically distinct. Archaea lack peptidoglycan in their walls, which allows them to survive as extremophiles in lethal environments, like the boiling geothermal hot springs seen here. From the domain level, the system filters organisms down through increasingly specific categories: kingdom, phylum, class, order, family, genus, until we reach a unique 2-part name, like Homo sapiens. This filing system functions as a biological map, tracing how millions of distinct species connect back to one shared molecular code. We have traced life from microscopic chemical pairs through mRNA scribes and protein factories, down family trees via alleles, and out to the classification of millions of species across 3 domains. What looks like biological chaos is actually a perfectly organized system of inherited data. Modern labs use automated machinery like this to sequence those genomes in real time, allowing us to identify mutations and develop cures for genetic diseases. Biology is the ultimate instruction manual of our existence. By learning to read the blueprint, we finally begin to understand the story of life itself.